or if you just want to support us. If you just want to support us and join our group, that would be amazing too. We're working on social events and educational events. We're going to do something like this every other month and do some networking, maybe some like clinical rounds, as Dr. Johnson wants to try, and really try to get the conversation going around what our patients are trying, what are they having success with. I think we'll find that we all share similar challenges in and around the space. So thank you again, everybody who's here, and any love, I appreciate it. You guys are great. And I'm going to kick it back to Leia. You can do an introduction. I have a little picture of my mug there.
Great mug, great mug. Yes, we will get started. And I just want to thank you all for attending our presentation. Just like Cody said, we will be doing more of these for PCCC. And please feel free to always send us, we will have information again, we'll post our information at the end, and it will include ways to reach out to us if you have any questions, general or otherwise. So we're gonna go ahead and get started. It's about 3 minutes after the hour, so I think it's a good time. So the way that this is gonna work is, what we're gonna do is, if you have questions as Cody is Dr. Peterson's presenting, all you need to do is either send it to the chat or to me directly. It's probably easier for me to see it if you send it to me directly. And then what we will do is, as Cody is going through, he will be notified that there are questions, and we will stop to make sure to answer questions. Our biggest focus about this presentation is we do want it to be somewhat interactive to allow you guys to be able to ask questions as we go. So that's how we're going to handle this. And without further ado, I would like to present Dr. Cody Peterson. And a little bit about Cody, just for those who don't know him. Cody is a pediatric emergency room pharmacist. He has his master's from the Masters of Cannabis. He has his master's in medical cannabis science and therapeutics from the University of Maryland School of Pharmacy. And he is an associate professor at the UCI. And he's also a the chief science officer for the CNIGMA, and he is on the board, founding member of the Pharmacist Cannabis Coalition of California. And one last plug for the Pharmacist Cannabis Coalition of California, for those who are not here when Cody mentioned it earlier, we are this presentation will be recorded. And everybody who is a member will have full access to any of our webinars, any of our past recordings, any of our CEs. So please join if you'd like to be able to have access to this. It is $10 per month for non-students, and for students, it's $10 for the year. So we really encourage you to join, become a member, especially if you're interested in cannabis and want to learn more, or want to help us help patients get the medication that they need. So without further ado, we have our wonderful presenter. Cody, take it.
Thank you, Dr. Johnson. I hope that everyone can hear me just fine. I am excited to be here. I was a little nervous putting this presentation together about synthetic cannabinoids. And the reason is, this is an emotional topic. People feel very strongly that synthetics either are the future or aren't the future, or they're good or they're bad. And I'm here to try to bring the steam out of the conversation and say that maybe this isn't even the right words. Maybe we're all, because we're not speaking the same language, we're running into issues around what we feel is right, wrong, safe and unsafe, moral and immoral. So thank you so much. Once again, I do have my master's degree in the topic, as do you, Dr. Johnson. But I'm not an organic chemist. There's a lot of chemistry to cover here, and it's very nuanced. So if you hear me say something a little bit off, ask a question. I'd love to follow up. We're all learning here together, but I hope you take something away. So we already did all that. We kind of have to look at the big picture real quick. I thought that this infographic did a nice job of breaking up the sort of cannabis science into sequences. So we got historical use. It says 2700 BCE, probably goes back 8,000 to 10,000, even longer potentially. And cannabis was all over the world for a really long time. Then in the 1830s, we sort of rediscovered it. The West, Dr. O'Shaughnessy was caring for British royalty in India, and he discovered cannabis medicines. brought them back and integrated them into Western medicine, where they took off immediately. One thing this infographic doesn't do is it totally skips over the medical applications that was going on between the 1830s and the 1940s. But we'll move past that. But I've done other talks for the SCC and other organizations about that. Now, we went from cannabis research in the 1940s through the 1960s, Dr. Raphael Mechoulam, Dr. Adams. And what happens is we move into cannabinoid research because we start to discover what this molecules actually looks like. And soon we discover how and why it behaves and interacts with the endocannabinoid system, which was amazing and perhaps one of the greatest breakthrough discoveries of the last 50 years. Moving beyond that, we start to figure out how to manipulate these molecules. And that's where we start to understand the endocannabinoid system, thanks to the help of synthetic cannabinoids, and we'll get there. And really into the 2000s, we moved into this age of drugs and medical cannabis. Medical cannabis has been legalized in numerous states, California, Colorado, et cetera, et cetera. We've moved across around the world. We've got FDA and Europe drugs approved all over the world that are made from cannabis medicine. So we've come a really long way from prohibition in the 1940s, but we've still got a long way to go, I think, to pay homage to the rich history of this plant. But that's not really what we're here to talk about. We're here to talk about synthetic cannabinoids specifically. So a little of this more modern conversation. Cannabinoid's a weird word. Cannabinoid meaning a molecule that's cannabis or cannabis-like, but really we almost mean that by THC, because THC is the cannabinoid that helped us discover this system and sort of biased the word towards CB1 and CB2 activity. And that's generally what we have going on. We have a molecule cannabinoid defined as a molecule that interacts directly or even a little bit indirectly with cannabinoid receptors, oftentimes modulating neurotransmitter signaling. Now, we also have molecules that share certain chemical structures. So for example, CBD or CBDV has very limited activity at the CB1 and CB2 receptors, but structurally it falls directly into the category of cannabinoids. So even that is a mixed definition. So types of cannabinoids, we're going to dive into this. So if Kenzie Ribolet is here, please hold off. I promise we'll talk about your paper later. Endocannabinoids, phytocannabinoids, and synthetic cannabinoids. You probably already know what this is. You can see I do have a little formatting error. That's what happens when you go from Canva to PowerPoint. I apologize. But we do have these endocannabinoids made inside of our bodies. All of us, every animal on Earth with a spine has a bunch of endocannabinoids and endocannabinoid receptors. We have phytocannabinoids, which are cannabinoids that come from plants. And generally speaking, we say cannabis, but we've learned now that there are cannabinoids coming from other plants, rhododendrons and rudellas, and all sorts of really cool insights that we've learned in the last 15, 20 years. And the reason we learned them was because of these synthetic cannabinoids at the bottom of the screen. You can see they were designed and produced by chemists, scientists, really bright individuals trying to help us unravel this mystery of the endocannabinoid system. And frankly, they've done quite a job in doing so. But as we'll get into, they've done more than that. Now, speaking of those courageous, brilliant minds, they all named themselves into the history books with the molecules that they've created. So the synthetic cannabinoids, at least historically, were sort of based around the structure of THC. So delta-9 THC here on the left, it's a molecule you're probably pretty familiar with seeing. I'm going to go to my little laser pointer here. So delta-9 THC is over here, and this is the molecule that technically the plant makes THCA, but the molecule that is considered a phytocannabinoid. Now you can see HU210, one of the earliest developed synthetic cannabinoids, has a few structural features that makes it quite impressive. And HU means Hebrew University, and Dr. Raphael Mechoulam's team at Hebrew University and around Israel were working on these molecules and developing them. You can see that at Clemson University, JWH018, this is a molecule that I first learned about in 2007, right as it was starting to hit the streets as a drug of abuse in head shops and things of that nature. And I learned about, this is John W. Huffman's molecule. This is one of the most prestigious and prolific organic chemists of our era. And he developed hundreds of these. This is his 18th molecule of this class to have developed. And something similar is going on with the numbering at Hebrew University, although not the same. Now, CP, Charles Pfizer, yes, that's Pfizer, the drug company. This is the name of the not the chemist, but actually the president of the company, who was the founding chemist. And then Alexandros Makrianis is another prolific organic chemist, medicinal chemist out of Northeastern University, and he contributed to the development of numerous of these molecules. So we've got a very clear man-made molecule, so man-made that we've named them after ourselves or after the institution which made it. So we invented synthetic cannabinoids, and these were a catalyst for learning, but also a conduit to drug misuse and abuse, if you like that word, but I prefer misuse. Now, if you go back, there's Raphael Mechoulam isolating THC from cannabis in 1964 in the same university, synthesizing HU210. Here's the JWH I mentioned by Dr. Huffman. And only 9 short years later, although Dr. Huffman, who was in the publication of this paper that we're reading right here, had no idea that these were going to turn into drugs of abuse. And that wasn't the plan, but he did develop synthesis instructions and very clear guidance around what to do, what to watch for, what sort of chromatogram we'd be looking at. So inherently science led to this. And that means open science, right? If this science would have been closed off because these drugs were far too dangerous, they would have never slipped into the black market and become a drug of abuse. But they did. And through the 2000s, spice was really taking off. You can see that first synthetic cannabinoids found in spice in 2008. We were starting to look for it. You can see very swiftly Europe withdraws, but we see all sorts of problems coming out of Europe. We see spice gold and other spice products creating psychosis and withdrawal, people falling over at school. We see the introduction of a word you may have heard, spice, called K2. These are common just market names. And you could purchase K2. And I recall I was at one time a young man in these head shops. Any of the smoke shops or places where you might buy glass, and all the time these products, which I don't have any pictures here, there's plenty of good talks on the internet about this, they always say not for human consumption, wink wink, because this is indeed being sprayed with synthetic cannabinoids. It's usually a plant product, dandelion, just for example, although that's not the product, sprayed with synthetic cannabinoids, and then you can smoke or burn it like you would other drugs. So why create synthetic cannabinoids, right? Well, I get that question, and it resonates with me a little bit too, especially if you think about what we did with synthetic opioids like fentanyl and how we got there from the poppy plant. But I will also point out that these are tools of drug discovery, and really important ones at that. They help us understand what receptor does what. So by introducing a drug that has selectivity for a CB1 receptor, for example, and doesn't interact with the CB2 receptor, we can learn the functions of that CB1 receptor. And the more selective a drug, the more we can isolate its effects to that receptor system. So for that reason, it was very helpful, particularly in the context of knockout mice. So we could really make it very clear what the effects being induced by the drug were by getting rid of extra proteins that the drug could interact with, and also developing the drug to be highly selective. Once we made selective drugs, we can radio label them with isotopes, send them through the body and brains of humans and or animals, take pictures of those radioisotopes, and now we know where those receptors are. Another critical part of understanding the endocannabinoid system you're so interested in. This is part of the science. And this is techniques used across pharmacologic discoveries and systems throughout modern pharmacology. Now, to reduce side effects or enhance activity. So humans think they can make something better. And the truth is THC, albeit a pretty useful substance, it does have a handful of side effects, ones that some people, particularly our government officials, feel they need to keep away from us to keep us safe. So too much psychotropic activity is considered to be dangerous or negative. So developing drugs that are not intoxicating or impairing, but provide the same analgesic or pain relieving effects or focus effects or anti-autistic behaviors in pediatric patients, all of those things we want, without the bad. And this has always been sort of the golden chalice or the chase of pharmacologists and medicinal chemists is to improve upon the science that we know by manipulating molecules, making them patentable drugs and making them things that can be improved upon and eventually sold at a profit, or for the betterment of humanity, although unfortunately we lean towards the first one lately. But you can't patent nature. At least it's not easy to do. And so for that reason, a lot of plant molecules do not get the same amount of research funding, focus, and attention that they deserve. Now, I'm going to take a breath here and see if anyone has any questions. We're soon going to get into a little bit of the nuanced language, and I think that's probably the part I'm most excited about. But I want people to appreciate that just because these are synthetic cannabinoids doesn't make them bad. However, they are indeed were designed, or at least originally, to mimic THC and to do it sort of more strongly, more selectively, more potently. They were never intended to be taken by humans. But that's, of course, what ended up happening, or at least not without proper study. I shouldn't say that. Of course, they would like to get them to humans, but not without proper FDA sort of preclinical and clinical research, which is not what happened with synthetic cannabinoids. Now, what we learned firsthand in the field is that these drugs are pharmacologically different, and I'll get to that in just the next slide. But this is because these are full agonists, and this is a pharmacological concept. If you're not familiar, I'm going to teach you about. But this full agonist, if you think of it as the receptor in our body, the CB1 receptor, as instead of a lock and key, as you probably do, borrowing this from Miyabe Shields, a brilliant educator, she said that think of it like a light switch or a dimmer switch. And yeah, the light can be on, but it doesn't have to be full blast. And if THC has your dimmer switch set to 50 or 60%, that's all the further it can go. But these full agonists go up to 80, 90, 100%, fully activating your light switch, your receptor, your endogenous cannabinoid receptors. And because of that, we get stronger effects and potentially more severe toxicities, side effects, syndromes, etc. Now, even though most of the drugs were designed to mimic THC or to activate these cannabinoid receptors, there are drugs that were meant to block it. Now, that sounds sort of absurd for anyone who likes to stimulate their CB1 receptors with THC, but the idea is that if we block the cannabinoid receptor, we can maybe bring someone back from having too intensive an experience, sort of like naloxone serves in an opioid overdose, something like that for THC, although THC overdose is not basically ever life-threatening, and as such, the need for the agent would not be as severe as, say, fentanyl, which is killing 100,000 people a year. Now, these blockers, rimonabant was a drug that was actually patented in Europe in 2005, and it was removed from market within just 1 year of its approval. And the reason is that this drug was sort of doing the anti-THC effect, and it was making people sort of deal with depression. They were dealing with more suicidal ideation, and unfortunately, it was becoming very severe. Well, I guess fortunately the drug company took it off the market. So there are no cannabinoid blockers on the market, but there was for a brief time in Europe. That was a synthetic cannabinoid developed very intentionally by medicinal chemists and then studied through this process I just discussed. So here's a little picture of what I just described. So remember I mentioned naloxone. That's an antagonist at the receptor. Believe it or not, this is what a receptor looks like, a 7-transmembrane receptor. And G protein coupled the receptor if you want to get super fancy. So this antagonist is blocking. Here's your receptor site, your dimmer switch, your light switch, whatever we want to call it. This is going to go ahead and sit in that little lock, but it's not going to activate it. It stops the cellular signaling machinery, okay? So this is what happens when opioids activate the opioid receptor. Right here's an agonist, so let's pretend this is the opioid receptor. They induce an effect, larger stimulus to signaling machinery. And in opioids cases, we get respiratory depression and pain relief. Both of those things are well-known effects. So when a patient overdoses and I introduce naloxone, an antagonist, it bumps it off that receptor site and then sits there. That's what also rimonabant was doing. And it was sitting there knocking THC off and actually sort of activating the receptor in an inverse way. So agonist is what we're normally dealing with when we're dealing with drugs, and synthetic cannabinoids fall into this far left-hand category. Full agonist, as you heard me say on the last slide. Now, full agonist is still a bit of a abstract term, but appreciate that it's more than the third one over, called a partial agonist. And this is a drug where small stimulus to signaling cellular machinery, regardless of the dose. So you can't really overcome the partial agonist. The shape of the key, the shape of the THC, which is this, let's pretend, is the green molecule, because THC is a partial agonist at CB1 receptors. The shape of the key is smaller and it only fits in this certain way. But if you look over here on the left, the full agonist, the shape of the key is different. And there's more potentiation of the effect. And this has serious implications. One more thing, because I don't leave any stone unturned. Over here on the right, you'll see allosteric agonist. I don't like that term agonist, but allosteric modulator is the term that I prefer. And this is a really important concept. This is a way that a molecule can work, say CBD, to sort of potentiate your endocannabinoid signaling, but not directly activating the receptor in your CB1. So an allosteric agonist is not working directly on what's called the orthosteric site or the primary light switch. It's sort of the key box on the side of the house that you have to turn on. And when you do, the lights on the inside are more sensitive to changes, or things on the inside are more sensitive. So this is an augmentation. This serves as a fine tuner at receptors, and allosteric agonism or allosteric modulation is another pharmacologic concept that does come into play when we're talking about cannabinoids, particular CBD. Now, moving to the next slide. I know that was a deep dive into pharmacology. I hope you were walking away, but you're not done yet. So we talk a lot about affinity when we're talking about drugs, and I put together a couple graphics sort of explaining these concepts. So receptor affinity is simple as thinking as how strongly is a molecule attracted to the target molecule or to the target receptor. Now, that receptor, remember, can be that light switch. That's sort of an aside. This doesn't have to do with the percent that the light gets turned on. This is just how much does the molecule want to be there, not what it does once it gets onto the light switch. And you'll see here that THC has a much higher, although the number is lower, the much higher affinity for CB1 receptors than CBN. And you experience this when you consume THC or CBN in that intoxicating factor. It would take a much larger dose, according to this estimate, and this is not necessarily accurate, but this would take 20 times more CBN to produce the same amount of action at the CB1 receptor, the high, that you would by THC. So that's a really impressive and I think very straightforward way to think about this. This is one factor of drug potency. Now, another factor of drug potency is actually known as efficacy. And this isn't a confusing word because it's used in other ways in therapeutics, in pharmacy and in nursing and in medicine. But this means what the receptor does at the light switch. Remember, we talked a dimmer switch. That's what we're talking about here. Now, the zero is the dimmer switch turned off and all the way down. Okay, we can turn that light on, we can turn that switch on with a full agonist, and we'll turn the dimmer switch all the way up to 100%. There's HU210 again, one of the first synthetic cannabinoids ever invented. However, when we stimulate with the right phytocannabinoid, probably, if you're listening to this, your favorite one, if we stimulate your receptors with T8 delta 9 THC, we only get a 50%. That's an estimate. That's a pharmacologic concept, but we only get a partial activation of the receptor and of the pharmacologic response. Even if we give more drug, which is what's happening at the bottom, we're getting increasingly more drug, but you can see the line levels out. Now, when we introduce something called a neutral antagonist, this is equivalent to naloxone on the opioid receptor. This is AM. Remember, well, I'm going to screw up his name, but the doctor from Northeastern. So this is a antagonist, a blocker at the cannabinoid receptor. And when it engages with that light switch, it doesn't turn the light on at all. It doesn't move it. It just blocks it, protects it. It kind of shells it off. This is different than rimonabant. And this is likely why rimonabant, the drug that was mentioned earlier, was such a problem on the market, because it looks like instead of just blocking the effects of, say, THC or of your endocannabinoids at the receptor, what rimonabant was doing, it was actually taking the light switch and going down with it and producing the inverse effects of THC. And for anyone who enjoys THC, you can imagine that wouldn't be very fun. And that was the general consensus among patients as well. So I hope you understand pharmacology a little bit better now. And if you have questions, make sure you ask them to me at the end of this talk. So there's another little formatting problem, but CBD-derived products are making headlines. And it did not take me long to find these. I didn't pull up the dirt. I knew about these off the top of my head. Okay, so let's look at the top left. Delta-8 craze concerns chemists, 2021. True. Okay, I've spoken to numerous organic chemists who are very concerned that the current state of regulations and the current way the law is written is putting patients and consumers at risk. And I mean that more than one. Good friends with no stake in the game other than like, come on, y'all, like we can do better. Now, on the right, police in the UK warn cannabis sweets after woman in 23 dies in East London. Okay, what the hell is a cannabis sweets? Okay, what actually happened is if you just go one more line down, and this is April 2022, by the way, this is just a year ago. A woman died from taking a Trilly Peach O that was tainted or spiked or laced or whatever word you want to use with synthetic cannabinoids. Okay, and this 23-year-old woman, and otherwise somewhat healthy anyway, passed away because of this synthetic spiked product. Now we have this mother charged with murder in, I believe, the Carolinas after her 4-year-old ingested a bunch of Delta-8 gummies and she didn't take him to the hospital. I think her primary defense was she thought it was CBD. And I wouldn't doubt that product said some CBD on it. People are confused about the language we're using. What is a synthetic? What is natural? What's legal? What's not? It's confusing, and it's all being blurred together by the media and by even the scientists. And then the FDA says, hey, this is dangerous stuff. And they don't really address the problems, is that it's dangerous because they won't legalize marijuana and they won't regulate Delta-8, but that's a whole different conversation. Okay, check this out. This is 2017, published in the New England Journal of Medicine, one of the most prestigious medical journals in the country. And this is synthetic cannabinoids continuing to increase in potency, a zombie outbreak caused by synthetic cannabinoid AMB furbenaca in New York. And AMB furbenaca is like a big, long, drawn out acronym that means some molecules here and some appendages there. We're not going to get into it. But what I'll tell you is when you see these big, long words and there's an F in there, furbenca or whatnot, that means it's fluorinated. You can see I'm circling the fluorinated ones. And most of them have F in the name, although not all of them. So the point of this graphic is this. Going back in history, Delta-9 THC, the only really psychoactive cannabinoid we had any significant access to for the last 10,000 years. Delta-9 THC has an efficacy concentration 50%, which you learned about, at 171. So think of like 171 as that's how much drug it takes to cause the effect, okay? Think of the effect the same across this whole graph here. As we start to invent more molecules, JWH018, and then ABD Paninka over in 2014, this number is getting exceedingly small. In fact, this one's exceptionally small. But the smaller the number, the stronger the drug. So we've developed all these drugs, and then they've moved into the illicit market. There's no good tests for them out there. They're being thrown into a bunch of products labeled not for human consumption. People are consuming them and having major side effects. Oh, I forgot how cute I made this zombie's eyes. The zombie outbreak is people activating their endocannabinoid system way more than weed does, way more than you would imagine. And they're walking around not behaving normally in an essential state of stupor or psychosis and not able to care for themselves. They might be vomiting, they might be talking to themselves. It's really not a positive experience when you talk to the vast majority of people who have consumed these substances.
Cody, before you go on, we actually do have a question. Thank you
Yay,
so
thanks
much.
for interrupting me.
Of course. Okay, so our question on was, what is the lethal dose of these synthetics?
It's a great question, and most of the time we don't know, because these drugs are not intended for human consumption. There are LD50 studies in rodents for some of them that we can extrapolate, but they're actually going to be all different. So for example, JWH018, you see how it needs almost only half as much as THC, versus this molecule, ABD Pinaka, this is 0.53. So let me do some quick math, 350 times more potent than THC. So the lethal dose of this drug is certainly going to be far less than the lethal dose of this drug, at least in a generalist sense. There are some exceptions. Did that answer your question? I hope so. Okay, so synthetic cannabinoid pharmacology, adverse effects, because the pharmacist can't get done with his talk without talking about the negatives or the concerns. This list is long, and you can see this is a 2023 review. I think it was only released in January of this year. So we have an established safety history with THC. Is it perfect? No. Cannabinoid hyperemesis is real. Impairment is real. The munchies are real. And so is, getting a little lazy. All of those are known side effects, but that's safe. We're talking about drugs that cause serious side effects. Let me point to the picture on the right. Agitation, confusion, hyperactivity, aggressiveness, schizophrenia, polydipsia, that's peeing a lot, that was a bad one, psychosis. These are common side effects of synthetic cannabinoids. Alveolar infiltrates, pulmonary infiltrates, pneumonia, hepatotoxicity, liver failure, diarrhea, abdominal pain, nephrotoxicity and liver failure, rhabdomyolysis, where the body starts breaking down its own muscles and causing eventual kidney failure. And then most particularly cardiovascular, because the same reason that THC causes tachycardia and causes the heart rate to go, these full agonists are cardiotoxic. And we know this, and we know that they can cause arrhythmias, MIs, hemorrhaging, all sorts of weird things with blood pressure, syncope, changes in pressure through the lungs and the heart that are not good, especially at high doses. And we don't know which drug you're taking. When you buy K2 or Spice, that's a generic term. It's like saying you're buying, I can't even, I'm not going to give you a good example, but it's a generic term. It doesn't actually mean the drug that you're consuming. It's just a representative of a synthetic cannabinoid. It could be one of hundreds. They've synthesized maybe even 1,000 of these molecules now. So it's really unclear, and it's not easy to say like, oh, this is that, unless you have even a test. These high potency drugs are poorly studied, inconsistently produced. They're sprayed on, oftentimes with little, like you can imagine spraying your plants with a mister. This is how these potent drugs are being sprayed onto plant material and then left to dry. They pose a significant risk to the public, and I wish the cat wasn't out of the bag. Now, you will hear about a couple instances of dangerous and life-threatening bleeding. Now, I was really intrigued by this because generally the endocannabinoid system, master regulatory system, does a lot of stuff, but has negligible or very limited role in blood stasis, hemostasis. And so I was really interested to find out why it was that bleeding could occur. And it turns out that some of these products, probably because they were contaminated during the manufacturing process, some of these synthetic cannabinoids have actually been tested positive for something called superwarfarins. And I'm not going to get deep into this. I had to go reading, but this is an extremely dangerous class of compounds. It is like warfarin, the medication we use for blood clots, often called rat poison. And this is something similar, but these are uber potent, long-lasting drugs that if you are exposed to, you will bleed and can continue to bleed for weeks or months. And it's highly threatening to your life. and you need a continuous administration of life-saving medicines. You're going to be hospitalized. It's a really big deal. And this is another example of a contaminated drug supply. And it's really unfortunate that we continue to prohibit cannabis and even other substances while, somehow letting things like these slip through the cracks.
Cody, we got another question.
Go for it.
The question is, are there any, capitalize any, demonstrable advantages to synthetic cannabinoids? And there's also a follow-up to that question.
Yes. And there are synthetic cannabinoids already in research, clinical research, FDA approved. And most of these synthetic cannabinoids are not the CB1 agonists that we're referring to, which is these drugs of abuse that we've sort of been hashing on, but they're actually tend to be CB2 selective agonists. So now they're going to activate the CB2 receptors, which are not located in our brain heavily, but rather primarily located in our immune system. And as you know, we've got a hyperimmune problem, as particularly in this country, but around the world. And that is particularly interesting. So there are definitely use cases for synthetics. There's a couple examples of some cancer fighting properties that could be super beneficial, because if you're trying to kill cancer, maybe it's good to turn the light switch all the way on, right? Maybe THC's little tickle at the receptor isn't enough, even at Rick Simpson doses. And so there are definitely conversations to be had. But again, we're talking about hundreds, if not thousands of molecules. And maybe not thousands, but 8,000. But the point being, at this time, very few have been studied in humans and have been found to be useful and safe, but more and more are being tested and more even being produced. When I was at the, which conference was I at? In San Diego, the Emerald Conference, talking to Colorado Chromatography, and they're doing some medicinal chemical work with their CBD derivatives and hydrogenated cannabinoids, and which is technically a synthetic, or I'm going to give you a new term for that later. And they're having really tremendous results against pancreatic cancer, something that we're desperate for therapies for. Really interesting. And it's thought to not be necessarily super psychoactive. So really interesting stuff. We have to learn more. But what happened here is science discovered and profiteers decided that, especially in the context of prohibition, I can make these drugs and sell them and people will buy them. So it's really unfortunate, but it's the state of affairs. So before we move on to the next phase of the talk,
one more question. Sorry, follow up to that question. So you've kind of gone over it, but just in your personal opinion, as a community, should we advocate for all and any cannabinoids, or only for those derived directly from the plant, or what other option you got? I added that last part.
That individual is going to have to wait till the end of my talk, and then we can address their question. Okay. So defining synthetics. So there's some definitions you kind of need to keep in mind as we move forward. These are mentioned a couple times. I tried to be consistent, but there's no gold standard for these words. So we're trying to put our arms around them. Look, natural products. Okay, this came from something living. It often represents the chemical makeup full spectrum, but not necessarily. Broad spectrum is probably considered a natural product. But ideally it's to represent that. Now, naturally occurring is something that can be found in nature. Okay, right? So this is all sorts of molecules. Pinene, right? We know the trees produce it, but it's also found in your cannabis. This is a naturally occurring terpene. Delta-8, THC, not common in nature, but does occur, has been found in untainted or just like, grown cannabis in tiny amounts, but it's there. THCP is something similar. That's tetrahydrocannabiphorol, and a 7-carbon cousin of THC. We found it in cannabis. It's on the market right now, big amounts. It's probably not coming from cannabis. And that's where things start to get really confusing. Naturally occurring, natural product, and synthetic all blends together. Semi-synthetic is a drug that uses compounds made from natural sources to make the final good. Okay, so this is CBD being converted into Delta-8. That's a semi-synthetic process. Now, this is sort of where did the drug come from, right? Semi-synthetic. Now, THCO acetate is another example. You take THC or CBD, just convert it to THC, then you acetylate it. Now you have this semi-synthetic drug. It's not a fully synthetic drug. It's not designed by someone. It's not named after anyone, note, right? It's that this molecule has been manipulated to have different effects. Now, synthetic, right, is a word that's getting thrown around a lot. This is the process of total chemical synthesis. This is a molecule made of other precursors that have not necessarily a plant source, oftentimes petroleum or other chemical processes, but sometimes plant extracts as well. Can make very novel products, right? You can build it. As a medicinal chemist, if you're going fully synthetic, one carbon, hydrogen, fluoride, fluorine, all of the things at a time, you can build this molecule, and that's what medicinal chemists are good at. And then another example or another word you need to appreciate is synthetically derived, right? So when THC is FDA approved, it's considered dronabinol, which is a very selective, a very particular structure of THC, which is one that the plant makes in abundance. But in the United States, under the FDA, dronabinol is not from a plant. Dronabinol is chemically synthesized. Now, that's different than in Europe, which it is chemically drawn out, but it's still the same chemical. So now it's so hard to try to put your hands on this because everyone says synthetic's bad and net plants are good. But what's a synthetic? And if it's a plant molecule and it's made synthetically, is that better it might be? So whatever it might be for that particular drug, those words that we're talking about, we need to be more specific. And that's really what point that I'm trying to make. Sorry if I rambled a bit there, but there are better words that we can use. So let's try to classify this a little bit, right? So let's talk about these naturally occurring compounds and these synthetic compounds, right? So those that occur in nature. You can't patent natural drugs, at least not very easily, although you can patent, there's a typo. You can patent FDA natural products under a certain regulatory pathway, although it is challenging. Synthetic and naturally occurring cannabinoids all have activity at cannabinoid receptors. They have those same functionalities. That's why they're in the class. But these synthetic cannabinoids, on the other hand, are not known to nature. We've never seen them in a natural sample of any product. Does that mean they don't exist? No, it doesn't. Nature is a big, wild, wonderful thing out there. You can also see that often these synthetic cannabinoids are designed, right, to either effect, they're trying to elucidate a certain effect in a rodent model or whatever, or they're trying to manipulate it on the street for legality. Because if you change the molecular structure of a molecule, because of the way our drug laws are written, that makes it not illegal again. And so there's been a cat and mouse going on between synthetic chemists and law enforcement. And it's unfortunate. I want to apologize again for this formatting. I'm sort of annoyed. I thought I went through all of it. But here's deltas. On the left-hand side, I put an example and an equivalent in the synthetic. So delta 9 THC is naturally occurring. It occurs in the plant. We can find it on lab analysis. A molecule you can find on the market right now, delta 7 THC. Yes, delta 7. You can find it on the market, but that is not naturally occurring. That's a synthetic drug. THCP, a potent THC derivative. That's a naturally occurring compound. We found it in the plant. THCO is not. It's never been identified in the plant. It's never been seen outside of a manipulated setting. Same thing, CBDA, found abundantly in hemp. Dr. Raphael Mechoulam made HU 580, right? So that's a CBDA methyl ester. That's a synthetic drug that's supposed to have these same effects. It's not intoxicating. There's no malice in the production of that one. But nonetheless, it's a synthetic drug. Beta-caryophyllene, a selective beta-2 agonist. Remember how I said some synthetics might be beneficial in that way? Beta-caryophyllene is a natural product. It has similar pharmacology, but you can't patent it and it's not well absorbed. So there's challenges. So then you make a new drug. So when you make HU 308, you're using the synthetic process to recreate the same sort of pharmacological effects as BCP, but now you do have a patentable drug, one that can be produced in a laboratory instead of in a plant source and all sorts of stuff like that. All right. So that's classifying, but not sourcing, right? Sourcing is different, but they use same words. Synthetic is a word to describe source as well. So fully synthetic, AKA, is produced via chemistry. This has nothing to do with plants. We have semi-synthetic that I already explained. This starts with some plant material, manipulates it into a product. And then, of course, you have biosynthesis, like cannabis itself, like the endocannabinoids inside of you. Those are biosynthetic endocannabinoids. So biosynthesis is happening, and we can use biosynthesis in the same way that we take sugar, feed it to yeast to make alcohol in your beer. We can now do this with genetic modifications to make cannabinoids, which is pretty cool. And we'll talk about that.
Hey, Cody.
Yes.
So my question for you is, what would commercial conversion of THC to CBN be considered? Semi-synthetic?
Yeah, I mean, so we're going to get to classification, and I'm going to have you we're going to ask you that question together at the end of the talk, Leah.
I'm just giving you more questions from people.
Oh, that's not your question. Yeah, I can't wait to explain it to you. You're going to get all the answers at the end of the talk. And thank you to Kenzie Riboulet for the hard science on the background that I'm going to teach you. So sourcing chemical synthesis. So this is one type of sourcing. We can do full chemical synthesis. We don't need organisms. We don't need plants or farmers or contamination or variants in the sunshine. That's all helpful. But you do need a smart chemist, one that knows what's going on, and then you need an analytical chemist to test it, because you can produce a lot of stuff. You can technically use some components from plants, technically. I want to point out that chemical synthesis is challenging to control. We're talking about things, I mean, we have a lot of control, but you can't manipulate the way the molecule moves in free space, which leads to byproduct production. You're producing things that are not your intended target. So if you're trying to make CBD, you might make irregular CBD, the one that's flipped in three-dimensional space. And that's not ideal. And it needs to be tested for and cleaned up. It can be cleaned up. It can be avoided, but only with skilled chemistry. And I'm talking about advanced analytical testing that is not affordable. But it's the gold standard in chemical synthesis, and these processes are well established in pharmaceutical companies. Like, this is my background in pharmacy, where I still practice today. There is very stringent controls on what drugs need to be made, what byproducts need to be tested for, what sort of policy procedures, SOPs, et cetera. And that sort of regulation does not exist in the hemp space. Now, lots of things going on here, but I want you to note that CBD is in the middle. And then depending on the circumstance that you put CBD under to isomerize it, to convert it into psychoactive cannabinoids, it can produce all of these different molecules. So some you've heard of. Look at Delta 9, our buddy. Delta 8. There's Delta 7 I mentioned earlier. This does not occur naturally in nature. We've got 1,5-dihydroxy Delta 9. Don't know what that is, but we know it's not natural. Down here, we've got something unique. If you notice, the structure of this molecule is called iso-THC. It is 3 rings, but it's kind of wonky. It's in a different position. And because of that interesting position, it has different pharmacology that we don't understand. How do we get here? Well, in 2018, the Farm Bill made growing hemp legal, and it was pretty popular. Farmers are looking to diversify, so everybody jumped in. Bunch of CBD came to the market because it turns out the supply chain for hemp isn't very good, and our varieties aren't quite right. So we weren't tuned up. So now we have a bunch of CBD flood the market. Price goes down. People are trying to figure out what the heck to do with these molecules, and they're trying to make a living. And they said, hey, they wrote this Farm Bill kind of funky. And if I read between the lines, or pretend I can't fill in between the dots, then it says only Delta 9 THC is illegal and everything else is free for all. So I can make all these molecules, and that's what this picture is from. These are all psychotropic cannabinoids. I can make all these molecules, sell them onto the open market, and probably make some money. So I know that Europe is a huge importer of HHC, Eastern Europe, Poland and Czechoslovakia, or that area, Czech Republic, excuse me. is HHC. A lot of that flowant. CBN is a common product in the hemp market, and it is technically psychotropic or impairing, although quite weak even compared to Delta 9 THC. Lots of room for interpretation and very little regulation. I don't know how else to say it. This chemistry you're looking at over here, it's not super easy. It's not super clear, at least to the layman or to the average bachelor's degree of chemistry, what to do there. Now, sourcing from hemp, we also have to consider that this process isn't clean. We're producing these isos and we don't know what they do. These have never been tested properly in humans, and they're not being tested properly in the laboratories many times. There's also some acids and some catalysts that can be lingering in the products, and we don't know what they do, especially not when they're vaporized. So there's a lot of concerns around what these products are, what's in them, could they be bad for us? And if you think about it, I told you about all that CBD. Well, I'm sure some of it is of low quality, and low quality into your synthesis will result in low quality out. So you're going to have some other stuff. If it wasn't properly pure CBD that they convert, then you're going to produce other sort of gunk in the background. Now, kind of deviating from traditional ways of producing drugs, this is a sort of modern way of producing drugs, and this is using organisms that are genetically modified. We do this for lots of medicine now, but it's only beginning to happen in the cannabis space. This utilizes genetic engineering to take the genetic information from, say, the cannabis plant or something very similar to that, splice it into this organism, whether it's a yeast or whether it's a bacteria. It puts the cellular machinery of the cannabis, it puts that THCAS enzyme in there, and we can produce THC, for example. And that's kind of what we're looking at over here on the picture that we did. Now, not we did, that this wonderful scientist put together, and I have the pleasure of sharing with you. So I want you to appreciate that when we're using enzymatic processes, which is what we're doing, we're introducing the genetic material that the plant has into this organism, that's folding that molecule just like the plant does. We don't have all this iso delta 8 and delta 7 production and all these weird funky molecules, because unlike chemical synthesis, which is sort of like molecules floating and hitting together and fusing, the enzyme sort of gathers everything it needs, it folds it up, and it spits it out in like a nice little tight package. So enzymes fold very cleanly, and chemical synthesis is sort of a messy process that needs to be cleaned up. This process of genetically modified production of cannabinoids, which I like to call agricultural biosynthesis. This can be really simple in the molecule, or the organism can just eat sugar, or it can be kind of complicated and you have to feed it sort of the exact precursors. So it sort of depends on your technology. One really big driver for this tech that's going to change everything is that it's super scalable. So once somebody really gets their process down, finds a market for their cannabinoids, it's going to be really easy to compete heavily with anyone who's producing cannabinoids in a farm. Of course, they won't be growing flower, so there will still be a market for cannabis, but it's definitely going to drive down the demand for growing at scale to produce isolates, like currently happened in the CBD market that led to the glut of psychoactive THC derivatives. Patentable. Lots of IP in this process, right? You've got the genetics that you're infusing. You've got, all the processes involved in keeping these organisms alive, reducing those molecules, extracting those off, and then packaging them out. So lots of intellectual property that can be patented, which essentially, puts these companies in a pretty good position. Okay. Now, for the question everyone's asking, the question of really where I set off to have this conversation. 2 years ago, I posted a great article, I thought, about synthetic versus endocannabinoids versus phytocannabinoids, and I got a lot of positive feedback, except from one individual, and that is the author of this study. This is Kenzie Riboulet-Zamuli. I'm sorry, Kenzie, if I screwed up your name. And I like feedback a lot. And I thought what he said was interesting. I acknowledged it. I looked at his paper. I thought it was too difficult. And I sort of dismissed him, admittedly. And on the, second or third time I'm having this conversation and nobody seems to understand the direction of the conversation and what we're trying to get across, I said, maybe I'm going to go back to this paper. And I've decided that I think this is the best way to go about this. This is the leading paper in this space. And this is what I'm going to propose how we go about this, at least when we're talking to each other, the insiders of the cannabis space. When we're speaking to individuals who are not in cannabis, we don't need to get into the nitty gritty of this. But let's talk about it. And the reason we need this categorization is so we can all get on the same page legislatively, regulatorily, so that everyone can be speaking the same language when it's important that we're speaking clearly, concisely, so that we don't find ourselves in another Delta 8 cluster or whatever is going on in the hemp space right now. Okay, so on the left-hand side, you'll see the two potential options for any cannabinoid. So paleocannabinoid, meaning natural, or neocannabinoid, meaning new or novel artificial compounds. Okay, so all cannabinoids will fall into one of those two categories, right? That was me a few slides ago. We classified our cannabinoids into naturally occurring or synthetic. Now, if you follow this chart along the middle, that's the wrong word, so sorry. If you follow this row across, you can see that at the top, we've got synthesis and biosynthesis delineated, okay? Biosynthesis means synthesized by life, as you might imagine. So that's the endocannabinoids in us and the phytocannabinoids in plants. But we have to accommodate this brewed cannabinoid, this yeast-produced cannabinoid, and potentially some other things that might happen into the future. Okay, so now we need to put words to those functions so that we can understand them. So when things happen naturally in the cannabis plant, it's biosynthetic. So that's true. It means the actual way it's supposed to happen. Biosynthesis. Okay, EU. Now, when this biosynthesis is happening outside of the original host organism, right? So this is happening inside yeast instead of inside the cannabis plant. That's called dysbiosynthesis. So this is abnormal or modified biosynthesis. And then we have the chemical synthesis that we mentioned before. And this is posynthesis, or sort of what means in vitro or test synthesis. So posynthesis is what it means to be outside and doing things fully synthetically or semi-synthetically. Okay, so let's follow this along. Delta 9 is a paleocannabinoid. It is a natural occurring cannabinoid. Everyone agrees it's got to go into row A or row B. So we're in row A. Then we follow across. Cannabis is the THC coming from the plant? Is it a phytocannabinoid from cannabis sativa? It sure can be. Can it be made in the yeast organism? I'm telling you that it can, and we're moving in that direction. It's definitely coming. So yes, it's also a dysbiosynthetic paleocannabinoid. And then lastly, when a phytocannabinoid is made through full chemical synthesis, it's called a posynthetic paleocannabinoid. Okay, same words. Let's go down to, oh, actually, never mind. I'm going to leave that off. So we've got the dysbiosynthetic when something's made in the other organism, the non-host organism. Biosynthetic when it's made in the original organism, and posynthetic when we have an actual chemical process. And THC will always be a paleocannabinoid because we know it occurs in nature. Okay, so this is the fun part where we're going to practice. And so I'm going to have you do the first one, Dr. Johnson. So we've got Delta 8 THC. Okay, so it's got to fall into paleocannabinoid or neocannabinoid. What do you think?
It's a paleo because it's THC.
It's a paleo because it has been identified, even if only in tiny amounts, in the cannabis plant, in nature. And as such, we can say that it is a paleocannabinoid. So now we can wipe off, this is not a synthetic, which is what neocannabinoids represent. This is a naturally occurring paleocannabinoid. Now, which synthesis is it created by?
Depends if you ask Mark or not. I believe I normally would gear towards the first one, the U biosynthesis, but in very minor amounts. But traditionally, I would say that it's usually synthetically made outside of the body.
I like that. It's over here in the phytocannabinoids obtained with starting material from cannabis sativa, semi-synthesis. That's a post-synthetic cannabinoid. I agree. Delta-8 is usually a post-synthetic, but it is a paleocannabinoid. It does exist in nature, but it's not usually produced in the plant. Certainly the products that are on the market are not used synthetic paleocannabinoids. They are post-synthetic paleocannabinoids. Okay.
Yeah, I'm with you, Mark. I'm with the Poe. Don't worry. I got you.
This is technically Kenzie who wrote the paper, but Mark probably agrees. Okay, Delta 7. Leia, we're going to do this one together, and then I'm going to ask the audience to chime in the next set.
Sounds great.
So Delta 7, what do you think? Paleo or neo?
I'm going to go with paleo, but I'm also going to go with Poe.
Okay, so Delta 7's never been identified in nature, as far as I'm aware. The plant only really folds Delta 9. It looks like Delta 8 can be naturally isomerized because it's a more stable version than Delta 9, believe it or not. But as far as I'm aware, Delta 7's never been found, which would then put us into the neocannabinoid area. I just jumped ahead, as you might see. Leia got the first one right, mostly post-synthesis or dysbiosynthesis, but there might be this little tiny production potentially in the plant. And then down here, Delta 7 is not. This is a neocannabinoid. It would have to be produced by post-synthesis chemically or by dysbiosynthesis, which if we can manipulate the genetics, all of a sudden the potential of what molecules we can make is enlarged, as opposed to nature sort of evolved to do what it does.
That's a very good point. I haven't really heard about Delta 7 THC, so that's a very good point. It's got to be a neo.
I mean, it exists, whether you heard about it or not. I think it's more about whether it's happening in the plant or not, whether we've seen it in nature.
Exactly.
Okay, so now I'm going to invite someone from our crowd to turn on their camera and participate. I would love to see your smiling face. So who's going to do it? Who's my glutton for punishment and do JWH018? Come on, somebody's got to have their makeup on still and wants to come on here. Do it. Do it.
Wonderful. And oh, don't forget, raise your hand and I can I'll say a name. Let me see if I can.
Okay, Vaughn, do it.
All right, I'm here. Let's see what we're looking at.
JWH018. Is it a paleocannabinoid or a neocannabinoid?
I'm feeling neocannabinoid.
Neocannabinoid, novel. Well, that's a good way to know because John Huffman's name is on it. So it's pretty clear that he made that one. Okay, so then tell me, do you think we make it in these organisms? Is it primarily made in yeast, or do you think it's being made in the laboratory?
I'm gonna go with laboratory because it's John moving with it.
Okay, so then now the toughest part, how do we say the name of this cannabinoid?
Theosynthetic.
I like it. Go. Yeah. Go for
Theosynthetic neocannabinoids.
Yeah, good job, dude. That was great. And you were right. I totally agree with your answer. But we can do some crazy stuff in genetic laboratories. I have no doubt we could figure this out eventually.
So now my question, right, is now once it reaches that laboratory, and let's say we start creating new derivatives from it, would it now go from JWH like 19 or 20 or things of that nature? Is that how?
That's not how the numbering works, but that is how the drug development process works. They'll find a molecule, they'll add an arm on it, and they'll name it another number, and then they'll test it, and they'll see what it does and all that. But yeah.
That makes sense. Cool.
That's medicinal chemistry, dude. That's what they do. These guys look at the molecule, they figure out which part of the molecule is important, and then they like, oh, I'm gonna put this there. I'm gonna move this way. I'm gonna move the double bond that way. And what is it in a rat? And then if it does cool in a rat, they're like, maybe we need to start looking at humans.
Got you. So that's even giving me a better understanding of like how this synthetic derivative process is even going, and like, you know, market ability for these new synthetics, because, you know, once a new synthetic reaches this market, we have that hold up. What is that? You know, when we start seeing those new derivatives, but then we start understanding why those derivatives even came about, it gives you a little better understanding.
Yeah, and how they came about. They synthesize what's called a series. So they make 50 new compounds and then test them all and look for the cool ones. And then they keep the cool ones and test them more. And the ones that, you know, killed all the rodents, they're like, maybe we don't want that one.
Right. That's cool. That's cool. That's fine. I appreciate this. And thank you for even giving me a chance to answer this. Thank you.
Yeah, sure. We got one more, THCP. Kayvon, we're going to have somebody else go. Thank you. Who wants to chime in? Dr. Santiago Valenzuela, come on up. You can just talk. You don't have to turn your camera on. But oh yeah, there you go. Go for it.
Hi, everybody. THCP, definitely a paleocannabinoid because it's been found in minute amounts on the plant.
Great.
And it's definitely a post-synthetic and could be a biosynthetic, correct?
Well, it can be all of them. And we know that because you said it's been found in the cannabis plant. I would point out that if the year was 2017, right? Like if this was 5 years ago, this is a neocannabinoid because we hadn't discovered THCP in the plant yet. So it's really interesting how the language evolves, but this helps us get there. This helps us really categorize better. Okay, I agree mostly. And I want to point this out for our audience. There's a lot of THCP on the market. I guess I kind of got to this earlier. It's there. I personally have bought some because I was like, what is this? Is it really going to be different? Right. And I have my friends try and like, is that irresponsible? Maybe. But the point is like these substances are being sold to kids. These substances are out there and like we need to be trying to wrap our head around it. So I asked my smart friends like Mark Chaldone and they say like, yeah, the THCP is bullshit. It's not. It's actually derivatives, and you know, it's not being extracted from the plant. So this is mostly a post-synthetic paleocannabinoid. So amazing. I'm so into this group right now. So I only have one more slide for you, and I want to just finish off strongly. If we're looking forward and we're trying to put our head, you know, sort of wrap our hands around this, we need clearer language to help people understand what we're talking about and where the substance they're consuming, where the substance they're purchasing is coming from. Okay, so the whole last slide is dedicated to that. Synthetics tend to carry more risks due to this potent pharmacologic effect. It doesn't have to. We can create partial agonists. There are some examples of that, but as a tendency, drugs that are being misused, the stronger sort of the better for the user, often trying to escape from their reality. So the fool ones sell better and they made it their way into the market. It's probably not the epidemic that we have going on with fentanyl, but it's definitely risky. But you know, this doesn't mean that this is all bad. This could still potentially lead to good medication, but we do need to be mindful of those serious side effects. We're going to see more of this. We're going to see more of it in the Delta 8 space and the derivative space. We're going to continue to see it until the FDA puts sort of some guidelines on this. But we're also going to see FDA approved. We're going to be targeting cancer and arthritis and Alzheimer's and GI disorders with synthetic drugs that activate and target parts of our endocannabinoid system. And we can't get away from that, right? Without tearing down the whole system. Likely to continue seeing the evolution of these street synthetics, as I mentioned. I really want to draw a conclusion here to fentanyl. Opium has been a problem for 2,000 to 5,000 years. Opium. Then we got to heroin, which was only invented by drug companies 150 years ago. And we had a real epidemic. People were dying, lots of them. And then we moved on to oxycodone, another semi-synthetic drug that caused a whole epidemic. And now we're on phase 3 where we have fentanyl and we have hundreds of thousands of people dying. And it's a fully synthetic, ultra potent opioid agonist. And what happened is they found morphine. If morphine's THC, they've made fentanyl, and we've made these. So we need to really be smart about this. We got to stop with this prohibition approach, because look what it's creating. And we need to be more thoughtful. We need to help people get access. That means home grow rights, full federal decriminalization. States can figure it out, whatever. Medical care for medical needs. We need to do better. And we need to have sensible policy that involves experts. We can't have legislators writing laws that don't have the right language. So this part of this conversation, everyone who's here, I empower you to use this language, especially when we're talking about policy making, because we need to be clear so that we don't find ourselves in this cluster again. I want to thank you so much for your time. I appreciate your questions. I hope I answered most of them in the review. I want to implore you to join. I am recently the director of new members, or something to that effect, some title. And so I am making a push. I hope that you learned something today, and I hope that you have questions. Thank you.
Thank you, Cody. That was absolutely wonderful. Just a reminder again, this has been recorded. However, we might be able to share bits and pieces of the recording, but the recording is a benefit of being a member of PCCC, and that's the Pharmacist Cannabis Coalition of California. You need to be neither a pharmacist, nor do you need to be from California to join. We welcome everybody. We are all inclusive. We love everybody. So please join. You get free access to all of our webinars and everything else. So we do have a couple questions, Cody. The first one was, so if a certificate of analysis shows THCA as the predominant cannabinoid in a product, is it most likely from the cannabis plant and not a synthetic?
Ooh,
That's
good question. So it's not that THCA couldn't be okay, let me back up. Is this Delta 9 THCA? Because that would be a big factor. It wouldn't be cost effective to manufacture Delta 9 THCA, because the plant makes it in quite a bit of abundance, and you can grow the plant at scale quite well. It's when you get into minors, it's when you get into less common cannabinoids that the scaling the plants doesn't really work, and they really lean into synthetics. So if the question is about THCA, my response would be it's likely plant derived. The flip side to that would be if this is Delta 8 THCA, it is absolutely some sort of derivative or synthetic. And I mean, you know, buyer be warned, or whatever you would like to say. I'm not going to say no, but note that it is synthetic, and synthetic isn't bad. That's not my take. My take is we need to be more thoughtful around the way we treat drug policy. Oh, and I think on my last slide, I overlooked it. I'm going to go back to it, or just read it out loud to you. I said, regulate drugs like drugs and plants like plants. And I actually didn't read this section of my slide, but like, we have to be more thoughtful around, hey, wait, if we're making drugs, if this is a what is it? Not you. What's the last one? Oh shoot, the synthetic, the fully chemical. I can't even remember the damn word right now. If it's fully chemical synthesis, maybe we should be treating it like a drug. If it's coming from the plant, this is eubiosis. It's in the organism. This is life. You don't get to tell me what lives and doesn't live. That is not the government's job. So that's my take.
I agree. And actually, to follow up on that too, just to give a little bit more insight on that question, the person is trying to figure out pretty much about how to really explain to their patients and consumers about what to get and what not to get. And again, every state's different, but what I can say though is that very often, if it is synthetically made in the dispensary, you'll see the words Delta. They rarely put Delta on products. Now, I'm not saying that they can't, but they rarely put Delta 9 or they usually rarely put Delta 9 THC on an actual THC extracted product. It's very rare. Not that it can't happen. It's just very rare, because dispensaries don't want the word Delta. They just want to say THC. It has THC in it. So that's usually a really good clue. If you see Delta, I would question where it came from. The other thing is also, if you see Delta 8, it was synthetically made. You're not going to be able to extract Delta 8 from a plant to get it into the milligrams level that you're seeing in these edibles and gummies and so on and so forth. And also flower, you can't always assume just because it's flower, it's natural. And actually, Cody, I like your description of the flower. So you want to give them a little heads up on how THC, synthetic THC, or Delta 8 or Delta 9 THC flower is made?
Wow. So there's a little bit of both going on in the hemp space right now, right? So we had a conversation, you and I, yesterday around, this Delta 9 THCA flower that's being sold as hemp is just in flagrant disregard to sort of a clarifying point put out even around the Farm Bill. And it's selling marijuana under the hemp law. But there's a lot of flower out there too that's being marketed or is Delta 8 flower. And you'll find that this Delta 8 flower usually has a lot of Delta 8, but also a little bit of CBD. And this is because this is hemp flower that is first sprayed with terpenes to make it extra sticky. And then it's rolled around in some sort of either keef or more commonly Delta 8. Yeah, not keef in this case, sorry. But Delta 8 isolate. And then it looks sort of frosty and very potent. So I think you need to be weary of that. And if on that product we had regulations that made it say something to the effect of the post-synthetic Delta 8, now all of a sudden everyone would know what was going on here, right? And is that the solution for everyone? Maybe not necessarily. Maybe we can just make better drug policy that doesn't make the consumer understand this. And if we wrote these words into the drug policy, then we could have more clarity on what should be on the market. And I think that was my biggest takeaway point from this talk.
Definitely. And one of our viewers said gross when you described how to make Delta 8 flower. And personally, that's my thoughts about it too. I figure, CBD flower on its own can have great medicinal benefits without inhaled by itself, without adding anything. So that's my go-to if you're in a state that can't get THC dominant flower. That's just my 2 cents. All right, well, let's see. Is there any other questions, any last minute questions? If anybody wants to go ahead and ask anything directly, feel free to raise your hand so we see you. Oh, we got Megan. Go for it, Megan.
Hey.
Hey, girl.
A question with the synthetics, like when people have and spice and everything, when people have died, like do we know what exactly is the cause of death? Is it the cardiac toxicity? Is it the bleeding? Do we know what exactly?
So again, K2 spice is a generic term, right? It doesn't even describe the drug people are taking. And every drug, the dose and the individual pharmacology is going to make a difference. So ways we've seen death is intractable seizures, status epilepticus, heart failure, MI, and just in general cardiac arrest. And we've seen people do like jump in front of cars and like psychosis. And then there's also the potential for kidney and liver failure, which could eventually lead to death, but as you know, is just terrible. So lots of mixed things, because the endocannabinoid system is everywhere. Activating it potently has a big potential, and it's going to depend on the unique pharmacology of the drug, believe it or not. And each one's different. Every name I listed could be a grossly different molecule pharmacologically.
Thank you.
Yeah. Go ahead, iPhone Zez.
Hey there.
Hi.
So we're involved in supplying clinical trials with all of the above that we've been talking about. So I'm just curious, what do you think in respect to clinicians using synthetics and investigations into disease states like COPD or other?
I'm hopeful. Let's get studying it, but we don't create new novel drugs and give them to humans. That's just not the way that we've decided we should ethically go about this. So I think there are certain exceptions, right? Like a compassionate use case, end of life, something to that effect that could be made. And I think that we need to do the proper animal studies that can get us to a point where we can start looking at novel drugs and their application in human disease. the FDA is there to regulate drugs. Their role in cannabis has been a complete nightmare, but generally speaking, their role in drugs has been a little bit better, a little bit. We'll kind of leave that off the table for now. It's like a whole conversation aside. But this is what they're good at. They know how to do this, and I think we need to do that to keep people safe based on just the side effects we're seeing, right? The things that I listed are not mild. So outside of a, compassionate use, I would say we need animal studies before we use true synthetics. Now, if you're talking about a paleocannabinoid that's derived via chemical methods, now you're talking about something totally different. Now you're talking about something that we do know has been exposed to people, and we do know that isn't going to necessarily have to go through the patent law and all that. So I think there could be an argument there, and we can do that safely. We can clean up these products. We can make true isolates and know that there's not crap in it, but we need to do that. Hope that helps, Zev.
Okay,
We've
thank
got,
you.
thanks, Zev. We have Misha as well.
Hey, Cody. So, question for you. Do you consider the enantiomer that's dronabinol to be it is synthetic, but would you call it a paleocannabinoid?
Yes.
Okay, because I mean, it does exist in nature. It's just at a smaller abundance than the plus minus.
So the plus minus is just it's like mixed, right? Like more like a
Yeah,
balance of
it's
those
plus
two.
minus, and the minus is the enantiomer that it's dronabinol, but that's purely synthetic. But it's not outside the realm of statistics that THCA synthase makes that enantiomer. In fact, it does. It's
Right.
just a question of that dronabinol is fully synthetic, but
Yeah.
it's structurally identical. So do we call it a paleo?
I would call it a paleocannabinoid, but one that is post-synthetic in nature.
Yeah. Okay.
And I'm sure that in their production, they're probably producing both of those, and they're probably cleaning it up because
Right. They're isolating one out. That's right.
Yeah, they wanted it for the patentability or the safety or whatever.
Well, yeah, but they're not doing it enzymatically. They're doing it purely
Right.
synthetically. Yeah.
Totally. That's why it falls into the post-synthetic side. But yeah, because minus still exists in nature, albeit it's not the primary version, I would say it's paleo. If we're going to say delta 9, right? The plant doesn't make delta 9 either. It makes delta 9A.
Yeah, right.
Go ahead, Zev, you're done. Anyone else have any questions?
We do have another one. The question is actually from your perspective. Do you feel any different about the exact same paleocannabinoids, whether it is sourced from hemp extraction, conversion from another cannabinoid, or grown from yeast? So in other words, out of those three under that heading, do you have a perspective on one versus another? I'm assuming they mean for safety would be my safety or efficacy would be my guess.
Oh God, can you restate the question one more time concisely?
Sure. So the idea is that for something, for example, delta 9 THC, do you find delta 9? Do you have any perspective? Do you feel any differently about delta 9 THC, whether it was extracted from, let's say, a THC plant versus made from a CBD plant versus made through the yeast?
Wow, that's really challenging. I give some of my opinion on the way that I think we should go, but this is even falling into preference. Look, I'm a whole plant guy at heart, and I think that's where I tend to fall. But if we're talking about isolated substances, if you're talking about THCA diamonds, whether it's made in a laboratory or made in a plant, if it's pure, whatever, then I'm pretty good with it. But that's the pharmacist speaking, right? I give out generics. I do. And I think you get to have opinions for yourself and make your own call. I think there's use cases for all of them. I think that they all have a place. I think full synthetic will have less of a role once we really master enzymatic production in that dysbiosynthetic pathway. But that's for the future and not so much right now. I think there's lots of roles for lots of drugs, and chemistry is not going anywhere. Isn't that right, Dr. Mark? So I don't foresee a change to these categories for some time, and I think they'll all have a role.
I just want to put my 2 cents in for that too. So for that, I really think that my only real big issue with the synthetic really comes down, derived from CBD, is how was it made and who did it? Because at the end of the day, it could be made perfectly well. It could be very safe and reputable and everything. But at the end of the day, with the CBD market, you don't know exactly where you're getting it from. So that's my caveat for the the hemp derived synthetics, is just, if you're going to use them, make sure you know where you're getting it from and that they know how to remove the acid that they use to make that product, because you definitely don't want to be ingesting that.
Yeah, and there's just the questions around like what laboratories are able to detect and what they're really looking for. And there's just not as much regulation. Although the legal marijuana or the legal cannabis market isn't perfect either. And we have to all work together to build a better industry. And I'm glad you guys keep showing up to learn and share. And that's how we're gonna do it.
Okay, I think we've definitely hit our full hour and a half that Cody and I did not think that we needed. So is there any last minute questions? Anything else? Anything? Just don't want to cut off anybody.
Share my
And
stuff, y'all. I appreciate you.
yeah, we really appreciate it. And PCCC, the Pharmacist Cannabis Coalition of California, we're going to keep coming out, like Cody said, with more webinars, more presentations, more talks. And we also are hoping to do a rounds too, because that is one thing that I personally am interested in, as I think it'd be really exciting to have a bunch of practitioners be able to get together and say, hey, I tried this for my patient, it didn't work. What would you recommend? Because I think the best we can do with cannabis medicine is to support each other and learn from each other. So Cody, I learned a ton from you today. Thank you so much. That was wonderful. And again, please join us at PCCC for more events like this. It was great having you all.
Thank you, everyone. I appreciate you. And we'll see you on LinkedIn.
Bye, guys.